processes of the reactions that you've got. So the deamination, the removal of nitrogen, and then the carbon skeletons as well. And then we'll do some nice clinical stuff at the end for you all, because there is some clinical medicine coming eventually. You will get there. So Socrative link is in the chat. Although, if you would prefer, if it's only one or a very small number of people, you can just type your answer in the chat box alternatively. What would you like to do, Nora? Typing, yeah, that'd be easier, wouldn't it? Cool, we will do that instead. Let me share Socrative instead. So question 1, tell me about the anabolic fates of steroids. And if you've got any questions, just type them in the chat. Or if you don't know, just put IDK. Yep, fab. So that's like the headline one. Amino acids form proteins. There's a few more things that they form as well. Can you think of any of those? Fab, hormones. Yeah, exactly. I will have a list for you here. So hormones is the other big group, so well done. So protein synthesis, some hormones and neurotransmitters. So you've got dopamine, adrenaline, your thyroid hormones, melatonin, and histamine. And you've also got glutamate, which is itself an amino acid, which is also the main excitatory neurotransmitter in the brain, and GABA, which is the main inhibitory transmitter in the brain. Cool. So worth remembering that kind of side note that they form some hormones and neurotransmitters and things as well. Question 2, can you name any hydrophobic amino acids? This could be some revision if you've watched the proteins lectures. So here we go. Hydrophobic ones: leucine, alanine, valine, and others. So the way I remember those is that they sound like names. You've got Lucy, Alan, and Val, and then there's the isoleucine as well. So serine, threonine, and tyrosine are your hydrophilic ones. And what's interesting about that is that those are the ones that get phosphorylated very often. If you're phosphorylating an enzyme or a channel or any kind of protein, chances are it will be on serine, tyrosine, or threonine, one of those three. And that's because they're hydrophilic, so they can accept that phosphate group. So next bit of kind of amino acid revision. Which amino acids are basic? The options are glutamate, histidine, lysine, serine, threonine, and aspartate. Yeah, fair enough. To be honest, this is something that you could cram the night before your exam. So that's not a problem. If I included an answer for this, there we go. Histidine and lysine are your basic ones. Those are your proton donors, and arginine would be the other one. Cool. And we'll whiz through these next few. So if you don't know, just don't worry about it. Which two amino acids undergo non-oxidative deamination? Do you remember this? Good. That means you've got something to learn from the lecture, because that's one of the main things that you've got to know about this. So without further ado, I'll get on with my slides. And by the end of the lecture, you'll definitely be able to answer this. So we're going to begin with the fates of the amino acids. They always talk about fates of things, which is very dramatic. But it's very simple. So we eat some chicken. The amino acids get taken up in the gut, and then they have three main fates. So they can be used for synthesizing the things like proteins and hormones that you mentioned. There's also a constant turnover going on. So all the time things are being broken down and built back up in the body. So that's another fate of amino acids. The proteins are constantly being recycled, and that's really important because sometimes they get misfolded. And so if we can't recycle them, that's a big problem. So we always need to be breaking them down and building them back up. And then finally, we've got catabolism. So we had anabolism at the beginning. We had the synthesis, and number one, what they make, the proteins. And we've got catabolism, which is the breakdown. And that'll be the main focus of the lecture. So as we discussed in the Succinate quiz, anabolic fates of amino acids, the big one is protein synthesis, but we've also got some hormones in there and some neurotransmitters. The turnover of proteins is about 300 grams per day, and the proteins are tagged for degradation by ubiquitin. And so the ubiquitin polymerizes, and that acts like a little marker that says to your breakdown machine, this protein needs to go. And so it gets snipped up into small bits. And so the amino acids are then required to rebuild those. Now, amino acids are also, when they're broken down, they release energy. So it's not just about getting rid of it if we've got excess, it's an energy source. It's not something that we store up like fat or glucose. We only use what we take in normally. However, there is a caveat to this. In the long term, when you're starving, if you run out of fat and if you run out of glucose, then the body decides that it does have a store, it's your muscles. So that's why they say that we don't store proteins for energy, because normally it's being used for something else. But in long-term starvation, your muscles themselves get broken down for energy. And this is one of the ways that people actually can die in long-term starvation, is that their heart muscle gets weakened. So let's get on to the meat of this topic, which is the mechanisms for breakdown of amino acids. And we're going to start with deamination. So the chemicals, amino acids, can't be directly deaminated, except for a few of them. And that's to do with what enzymes you've got, basically. So first, we have to get rid of that amino group, transfer it onto an alpha keto acid, and make glutamate. So glutamate is the amino acid that directly undergoes deamination. None of the other ones, except for glutamate, serine, and threonine, could be directly deaminated. And so the general mechanism for the beginning of breaking down those amino acids usually starts with swapping your amino group. This reaction is catalyzed by different aminotransferase enzymes. We might hear them referred to as transaminases. So different amino acids will have a different version. And these require a derivative of vitamin B6 as a cofactor, which is something you might get asked on a multiple choice question in your exams. As a particularly special case, you've got some branched chain amino acids, and you might see bodybuilders taking these as supplements, and that's because they go straight to the muscles. That's where they are degraded. They have an aminotransferase in the muscles that is required for the first step of breakdown of these particular amino acids, because their branched chain needs to be broken down first. So once you've swapped your amino acid, once you swap your amino acid into glutamate, it can be directly removed. But there's a couple of amino acids that don't need to have an additional amino acid group that doesn't need to be swapped, and that's because it's not in the main body; it's in the side chain. So this is another little exception. There's a few little exceptions here, and so these two are the ones ending in ene, glutamine, and asparagine, and they form glutamate and aspartate. So the amide group in the side chain is directly removed by hydrolytic deamination before you get the subsequent deamination. So those glutamate and aspartate acids, they've had their amide groups removed. So this is the second set of exceptions. You've got serine and threonine. So those are directly deaminated because they are those hydrophilic ones that we spoke about earlier in the quiz. So serine, threonine are hydrophilic amino acids. That's because they've got an OH hydroxyl group on their side chain. And this is a target for chemical attack. So a dehydration reaction can occur, and that allows the amino group to be removed. And this is the main funnel for all of the rest of them. So everything except for serine and threonine and that direct removal from asparagine and glutamine. This is the main funnel, oxidative deamination. Glutamate is the only amino acid that undergoes rapid oxidative deamination. Most of the other amino acids get funneled in, as I've said. Glutamate dehydrogenase is the enzyme that catalyzes this reaction. And as you've seen in the title, this one is called oxidative deamination. And so much like all the other kinds of oxidation you've come across, this will release energy. And the way that we pick up that energy is NAD. So to summarize our deamination, we have a few little exceptions. We've got the hydrolytic deamination, which are the ones that have the side chains that can be removed directly. We've got hydrolytic deamination. We've got non-oxidative deamination, which are the hydrophilic ones that can be attacked directly, and all the rest are oxidatively deaminated. So let's move on to nitrogen. So what happens once you've removed that NH3 group? Well, in its molecular form, NH3 is ammonia, and that's toxic to the brain. So we can't have that floating around in the blood. So what happens is it's turned into urea in the liver, and only a very small amount is left as ammonia and gets directly excreted. To reach the liver, the ammonia is carried in an amino acid. These are normally alanine or glutamine. So if you take blood from someone and you measure the levels of amino acids, alanine and glutamine will be very high. That's because those are the main carriers. They're suitable to be the carriers because they have, when you break them down, that turns into another stable metabolite, and then that can travel out again to pick up the nitrogen again. So it goes in a cycle. And the name of the cycle is the Cahill cycle. Apologies for the French in the diagram, but this had the best picture that I could find. So your glucose is made in the liver, or the foie on the right. That's where gluconeogenesis happens. And you don't get any gluconeogenesis in the muscle. So the energetic burden is all in the liver, and it's making all that glucose and sending it to the muscle. And then what happens when you break down glucose is you get pyruvate, which can be converted to alanine. And so alanine then travels back. And so I like to think of the Cahill cycle as having two kind of roles. One of them is that you recycle this alanine. This is more energy efficient, right? It gives you something you can use for gluconeogenesis. And the second one is nitrogen. So there's a net transfer of nitrogen from the muscle to the liver. If you look carefully on that left-hand side, where the nitrogen comes from is in the alanine. The alanine, any amino acid can give its amino group. So they've got those two roles there. One's for energy and the one's for nitrogen. So what happens when we get to the liver? So we've had our nitrogen transported in alanine or glutamine. It's come to the liver, and now it gets turned into urea. And the way this is done is by the urea cycle. So much like the Krebs cycle, we have a series of reactions that go round and round, and then an entry point for new metabolites. And just like the Krebs cycle, it was discovered by Sir Hans Krebs. And this was actually the first metabolic cycle to be discovered. If you're talking about cycles in essays, it's always important to start with where something happens. So this takes place in the liver, but specifically in the mitochondria, in the periportal cells. So what happens is your nitrogen comes in, it's combined with bicarbonate, and then it's phosphorylated, and carbamoyl phosphate is your entry point. It's the equivalent of your pyruvate in the Krebs cycle. So here we are in the liver, in the periportal cells, and the urea cycle is taking place partly inside the mitochondria and partly inside the cytoplasm. The carbamoyl phosphate enters on the left, and you can see this is an energy requiring process. ATP needs to be used up to make this. And then it follows round in the cycle, creating citrulline, argininosuccate, fumarate, and then urea. Another point is that the urea cycle is actually linked to the Krebs cycle through something called the aspartate argininosuccinate shunt. You don't need to know all the details of this. The main takeaway is that there's a link. Why is there a link? Well, there's a couple of reasons I thought of. One of them is that the urea cycle requires ATP, and the TCA cycle, the Krebs cycle, creates ATP. Another one is that the urea cycle uses up carbon dioxide, which is quite useful because that's a waste product of the Krebs cycle. So that's the nitrogen half covered. So to recap so far, we talked about the three different ways that nitrogen can be removed. The amino groups could be removed from an amino acid. Then we talked about what happens to them next, where they get sent to the liver. And now we're going to talk about what was left behind in the muscles or in the peripheries, the carbon skeleton. That's the rest of the amino acid minus the nitrogen. So the fates of the carbon skeletons, well, these are the bits that make energy. And they can either go into glucose or they can go into your fatty acids route. Now, something you will need to know is which ones make which. That's a very standard multiple choice question. And it's quite easy because glucogenic is basically most of them. Most of them make glucose. And they can do that either by turning into pyruvate or turning into other bits of the TCA cycle. What you do need to remember are the ketogenic ones, which are leucine and lysine. Those are only ketogenic. And then the mixed ones, which are isoleucine, phenylalanine, tryptophan, tyrosine, and threonine. So it'll be important for exams to remember which amino acids make glucose, which make ketones or fatty acids. You don't need to learn this diagram. This is just to illustrate the points, the exact points at which they all feed in. So in the top right of the diagram, we've got the green boxes, and those are some ways to feed into the Krebs cycle. If it goes into acetyl-CoA, that's mixed. It can go either onto acetyl-CoA, which turns into fat and ketones, or it can go back into the Krebs cycle. So that's a mixed portion. Acetyl-CoA, it tends to go up to fat directly. And all of these other ones feed into intermediates of the Krebs cycle. And these are far more common. So it's helpful for remembering because you only need to do these ones down here. So there are a few special carbon skeleton fates to remember. These are important in the grand scheme of amino acid metabolism, not just because they get turned into intermediates for energy, but also because they have really important roles in the way that nitrogen is moved around. So the first one we know is glutamate goes to alpha ketoglutarate. That's the reaction from oxidative deamination, and that's a really important one to remember because pretty much every amino acid will get funneled through that system, except for serine and threonine. Then we have the glutamine going to glutamate and asparagine going to aspartate. And then another one that is quite handy to remember is that alanine, when deaminated, becomes pyruvate. And that's really central to a lot of the carbon skeleton metabolism that goes into the glucogenic route. Quite a few amino acids are funneled into that. And it's also the reason, one of the reasons why we have the Cahill cycle as alanine. Right, let's get clinical now. So there are a few diseases of amino acid catabolism. Some of them are fairly rare and you are unlikely to come across them in clinical practice, but one of them is one of the most common metabolic diseases that you can have. It's still not particularly common, but as metabolic diseases go, about 1 in 10,000, so it's worth looking out for. So, with that in mind, let's look at a case report. So, a mother brought in her 1-year-old boy to see her GP with a rash. She is new to the surgery, having moved to the UK from South Sudan with her family 6 months ago. The GP notices that the child's head appears to be a little bit too small. The baby's been vomiting and is generally irritable. He hasn't said his first words yet and doesn't crawl around, and I'll tell you for free that a baby of 1-year-old should be able to crawl around and should have said their first words. So based on this history, a GP suspects something funny is going on and refers the infant for further investigation. So just type in the chat box instead of Socrative. What is your differential diagnosis, i.e. what diseases do you think the baby might have? What's going on? It can be as broad or as vague or specific as you want. If you've just joined us, we're just looking at a case report. We're having a think about what this young boy might have, what disease they might have as something clinical. So if you've got any ideas, type in the chat box. Could be genetic, could be an infection. Maybe some kind of toxin or insult. Kind of broad categories of disease we might think about. Let's have a look. So I came up with some broad categories that might cause this kind of picture. So a baby with developmental delay, and microcephaly, a small head. So we've got some genetic things. That's always a good category to go for if you're not sure what's caused a disease. So if you've been going to any of the genetic lectures, you might be able to tell me some chromosomal abnormalities. Another category, surprisingly for a metabolism lecture, we've got some metabolic diseases. So phenylketonuria, urea cycle defects, and mitochondrial disease. And it's those first two that we'll be looking at today. Another option is that the mother could have had an infection while she was carrying the child. Some common ones like that could be cytomegalovirus. So once you've got your ideas of what might be going on clinically, think, okay, what tests are we going to order to figure out what the baby might have? So a CGH array could tell you about chromosomal abnormalities. I'm going to take some blood tests to look at the metabolic ones. Intrauterine infection is a little bit harder to pick up from tests. So you would probably go for the history on that one and ask the mother, has she had any episodes of coughing and fever? And the results come back in this case, and they haven't got a genetic disease, which is good news. What they have got is elevated phenylalanine in their blood. So this child has got phenylketonuria. So phenylketonuria, or PKU for short, is an autosomal recessive disease affecting 1 in 10,000 births, which is fairly common for a metabolic disease. And it's a deficiency in an enzyme that is responsible for the transamination of phenylketonuria. So now we see why the transaminases were really important. If you can't do that first step, you can't break it down. That means that intake of phenylalanine in the diet hasn't got anywhere to go. And because phenylalanine is neurotoxic, the symptoms we get are related to the brain. So developmental delay we spoke about, which might present as a child not walking or crawling at the right stage. Seizures, vomiting, eczema. So our baby had a rash in the case, that explains that. The signs they might have: small head, pale skin and hair, and a musty odour. Pale skin and hair, I'm going to take you right back to the beginning of the lecture for the cause of that. Can anyone think why they might have pale skin and hair? I'll give you 10 seconds to work it out. Well done if you thought of it. So the reason why they have pale skin and hair is that melanin is one of the things that amino acids can make. It's one of those anabolic fates that we had right at the beginning. So we've got proteins, we've got hormones, and neurotransmitters. We've got melanin in there as well. Phenylalanine is involved in the synthesis of melanin, and so if there's a build-up of it, it inhibits the formation of melanin, and so the children will have pale skin and hair. The reason why we might not pick that up in the case is because they've moved from South Sudan, so it's not always obvious, that sign. Now, phenylketonuria is diagnosed routinely as part of the newborn heel prick test in the UK. So every child is given this test in this country when they're 5 days old. And the heel prick test is a panel of about 9 different diseases. Quite a lot of them are metabolic diseases. And the ones you can pick up very easily early on. They're treatable. And if you don't treat them soon, they have pretty severe consequences. So that makes them a really good candidate for screening tests like this one. Now, why hasn't this baby had it? Well, they've just moved from South Sudan, so a bit of a war-torn region, and so that's one reason why it might not have been picked up. A big kind of travel history early in life. So it's quite unusual in the UK to see a child with phenylketonuria presenting at 1 year old, because normally it would have been picked up before that age. Another set of metabolic diseases that I mentioned that this case didn't have. they could well have done, are defects in the urea cycle. So PKU was a defect all the way at the top of the chain of amino acid metabolism at the beginning with the transamination. Now this urea cycle defect is all the way at the other end. It's in getting rid of the nitrogen. As with most metabolic diseases, they're just deficiencies in enzymes. So if you want to think of a urea cycle defect, just name an enzyme from the urea cycle and put deficiency on the end of it, and you'll probably do just fine. So similarly, it leads to a build-up in the blood, this time of ammonia. And as you'll recall, ammonia affects the brain. And so that will lead to some very similar symptoms as we have with PKU, because coincidentally, PKU also is neurotoxic. Now, the treatment for these is no instant cure, as you can expect for a genetic disease. What we can do is control what proteins they're taking in, and therefore avoid taking in too much of the wrong kinds, and then put in the right ones. So, for an argininosuccinase deficiency, if you supplement a person with arginine, that can help. That can help push the equilibrium for that particular reaction a little bit further over, so they can try and remove some more nitrogen. And you also need to restrict their protein intake overall, so that there's not too much load placed on the cycle. And the reason why that works is because their deficiency, they might not necessarily be totally absent. And so supplementing them with some arginine will help what small function they do have in those enzymes increase. So what have we done today? We've placed amino acid metabolism in the really broad context. We've learned the reactions of transamination and deamination that deal with the nitrogen. We've then seen what happens to the nitrogen next. We've learned what the other part, the carbon skeletons, do, and which kind of different molecules they get turned into for energy. And finally, we discussed the common diseases of amino acid metabolism. So now if we go back to our questions, hopefully this time round, should be able to answer some of those ones that we didn't know at the beginning. So, which two amino acids undergo non-oxidative deamination?
I think we've got some answers in the chat, Jonathan.
We do, B, D, E, and F. Right, let's have a look. So, non-oxidative deamination. So it's the other way around. You're kind of nearly there, but then it's the other way around. So non-oxidative deamination. It's a bit of a funny one. That's a specific kind of deamination. It's not saying which. It's not saying these do not undergo oxidative deamination. It's a specific kind, and that's the one where they've got them in the side chain. They've got an OH group in the side chain that can be dehydrated and then directly remove that amino group. So there are two kinds of deamination that are not direct, and that's one of them, serine and threonine, which are our hydrophilic amino acids. So the next one, which two amino acids undergo hydrolytic deamination? So that's the other kind of indirect deamination. And our options are glycine, aspartate, asparagine, tryptophan, phenylalanine, or glutamine. So our answers for this one are asparagine and glutamine. So these are the ones ending in -ene, so they have the amine group in a side chain as well as part of the backbone of the protein. So that means that the side amino group could be removed directly before you get to the oxidative deamination. So asparagine and glutamine undergo hydrolytic deamination. So this is one of the main facts you need to take away from this lecture to be able to answer your multiple choice questions on amino acid metabolism, which is that serine and threonine undergo non-oxidative deamination, and asparagine and glutamine undergo hydrolytic deamination. If you can remember that, then you'll have the basics to answer a little bit of amino acid stuff. And the last little fact, which amino acids are ketogenic? And the answer for that one, leucine and lysine, the ones beginning with L, are the solely ketogenic ones. And then we have the mixed ones, which include phenylalanine and tryptophan. These are really big ones that have got rings on them, and so they have enough skeleton to go two different ways. Phenylalanine, tryptophan, tyrosine all have rings, and then glucogenic, everything else. So you don't need to remember those. That's easy. Fab. So if you remember that small number of amino acids and put those in those little pots, then you'll have everything you need for your multiple choice questions. Thank you for coming, everyone.
Thank you so much for that, Jonathan. That was really great.
Cheers, Harry.
I'll make sure that recording is